having to build up a solid bank of knowledge as a researcher. Knowing how to read journal articles efficiently in a strategized manner is probably one of the most important skills to become a successful scientist. Hello everyone, my name is Nick and I am a neurologist in training. I am also beginning my journey as a part-time scientist, and this is a channel where I hope to share the things that I've learned along the way. Today I'll be talking about how we can read journal articles more efficiently, beginning by focusing on one of the most important part of a journal article, also known as the abstract. I'll be giving you a framework to follow so that you can apply this strategy to each and every single article that you come across during your endeavor of becoming a part-time scientist. I'll be demonstrating the framework through an actual paper that I'm reading so that you can get a more practical grasp of the idea. Now without further ado, let's get into it. So here we see an article from Nature Medicine about Alzheimer's disease and how astrocytes play a role in amyloid beta, which is one of the proposed cause of Alzheimer's disease. And right below the title of the article, we see our authors, and below that would usually be our abstract. Now this is the standard layout for most articles within the scientific community. Usually the abstract would be a couple of hundred words. It summarizes the gist of the paper. And so it is a very useful entry point for us as part-time scientists to get an overview of the article, to decide whether or not we would like to move on to read the details. Because probably you can get around 80% of the gist just from the abstract itself. Then we can decide whether or not we would like to move on to read in detail. This really forms a triage process, as we like to say in the medical field, to discern whether or not an article is useful, whether we'd like to read it now, later, or store it sometime down the road. Diving into the abstract, it usually follows a very standardized structure, which is good because we can apply our framework to almost all of the journal articles out there. And this usually follows the layout of the entire journal article as well, consisting of four main parts. The first one would be the introduction, second would be the study design, third would be the results, and fourth would be the conclusion. So let's break down each of these four segments one by one, using this paper as an example. Number one, introduction would usually try to convey the why of this paper. Why was this paper written? How did the paper authors come up with this idea? It usually begins with a little bit of background knowledge surrounding the paper, as we see in this example as highlighted in red. I think this is a very nice demonstration as the authors explicitly stated the why, the questions that they are trying to answer. In this case, it is that an abnormal protein called amyloid has been proposed to be the cause of Alzheimer's disease. However, it is very interesting to know that a lot of patients who are asymptomatic, which means that they do not have any memory impairments, they do carry abnormal protein called amyloid. However, they do not proceed on to developing Alzheimer's disease. So why is that the case? That they do have the abnormal protein, but they don't have the disease? This is the knowledge gap that the researchers are trying to answer. Is amyloid actually the cause of Alzheimer's disease? Another piece of information that the authors have provided is that a certain trigger is needed in order to unleash the deleterious effect of these amyloid plaques, which is related to astrocytes, a subset of immune cells present in the brain. And so it is proposed that both of these aspects are required, both astrocytes and amyloid plaques are needed in order to trigger the downstream events causing Alzheimer's disease. Now, the second segment would be the methods, which explains what the authors did to answer the questions that they proposed. Here we see that the methods are highlighted in green. Here the authors explain that they used a biomarker study, which means that they drew blood from these cognitively unimpaired patients to see whether their astrocytes correlated with the abnormal proteins. Now, going on to the third section would be the results. And this is highlighted in blue. This is the section where the authors would summarize their findings based on the experiments that they've conducted. And in this case, the authors found that the abnormal amyloid proteins was associated with increased markers for Alzheimer's disease only in those individuals who also had positive astrocyte reactivity. So both of these components are required for an individual to develop Alzheimer's disease. In this example, the authors actually intertwine the second and third component, which means that part of this is actually the results as highlighted in blue. The authors then proceeded on to another type of experimental model, which utilized imaging to determine whether or not the subjects had features of Alzheimer's disease. Now, this imaging is called tau positron emission tomography. A mouthful, definitely, but what it basically means is that it is a scan of the brain trying to detect the presence of a protein called tau, which would suggest that the individual is suffering from Alzheimer's disease. The authors then stated the result of this imaging experiment, which is highlighted again in blue. They found that Alzheimer's disease, like pattern of tau protein accumulation, is only present in patients with both amyloid protein as well as positive astrocyte reactivity, which confirms the finding in the previous experiment involving biomarker plasma analysis. So here we need to be careful about intertwining of the second and third segment because it is only logical. First, the author states the experimental design, and then the result of the first experiment. Then the authors proceed on to describe the second experiment and the results of the second experiment. Last but not least would be our conclusion, as highlighted in orange. This is where the authors would consolidate what they have done, consolidate the findings of the different experiments, and come up with a conclusion. This would also fit the author's finding within the greater picture as provided by the introduction, and how this experimental results would again answer the question initially proposed. In this example, the authors concluded that their findings suggested that both astrocyte reactivity and also abnormal beta amyloid is needed to cause Alzheimer's disease. And so even if a patient does have these abnormal protein called amyloid, they may not go on to develop Alzheimer's disease if they do not also have positive astrocyte reactivity. It is only when both of these factors are present, then only a specific trigger for Alzheimer's disease would result. And of course, the significance of this conclusion is that it is important in future clinical trials in selecting cognitively unimpaired patients for Alzheimer's disease related experiments. Not only do they need to have the abnormal protein amyloid beta, they might also need to be tested positive for astrocyte reactivity to be qualified or to be categorized as a preclinical candidate for Alzheimer's disease. And so there you have it, 4 simple steps that forms the framework to approaching an abstract of a scientific journal. Now to recap, the 4 steps are: number 1, the introduction, which introduces the research question, the why. Second of all, the experimental design, how the authors set out to answer the experimental question. The third part would be the results, what the authors found through their experiments. And fourth would be the conclusion and how these results impact and fill the knowledge gap initially proposed. I found this to be a very useful framework in this beginning phase of my journey as a part-time scientist, because it really makes the whole process of reading journal articles much more efficient, and to help me get a grasp around the central idea of the paper, even if I decide to dive deeper into the details of the paper. I look forward to sharing more of these tips and tricks as I go on my journey, and I hope that these would help you along with your journey as a part-time scientist as well. And I'll see you in the next one.